Hello everyone. Thank you for joining me today for the first Poly user group meeting in 2020. I hope you're well wherever in the world you are. My name is Shefali Latwal and I'm a lead scientist at Elucidator. In this session today, I will be walking you through a live demonstration of Poly data lakes. I will be using COVID-19 multi-omic data lake as an example to show you what you and your team can use Poly for and how you can answer your research questions using the platform. COVID-19, unfortunately, doesn't need any introduction. As of 18th October 2020, there are close to 40 million cases in the world. Out of those, the total mortality has been over 1 million, so close to 2.8% of all cases. We know that the phenotype of COVID-19 can range from asymptomatic infections to very mild infections that resolve on their own in about 80% of the cases, to very, very severe infections that can actually be fatal. Severity of COVID-19 is currently diagnosed based on clinical characteristics such as oxygen saturation level in the blood, respiratory rate, and other clinical metrics that a healthcare worker or a doctor may determine. Now, by the time these symptoms are observed, the cases have already become severe. And that is a problem because mortality in severe cases of COVID-19 is much, much higher than the average 2.8% that we have observed. Therefore, it's extremely critical to identify which cases have the potential to become severe. And if we're able to do that successfully, it'll allow us not only to implement prevention and control measures in a timely manner, but also to prepare our health systems to provide supportive care in an optimized manner and hopefully achieve better outcomes. We know that today disease severity of COVID-19 has been linked to several risk factors. We know that age is a risk factor, gender, ethnicity, as well as certain comorbidities such as diabetes, obesity, cardiovascular diseases are also risk factors, but there are no definitive markers that can say whether a patient is likely to become severe or not. Therefore, a very, very important question for the research community is, can we predict the severity of response to COVID-19 before the cases clinically become severe? In other words, can we take existing COVID-19 data of patients who are severe or non-severe, and can we build predictive models based on certain characteristics? And can those characteristics can then be measured in the clinic to predict whether a patient is likely to become severe or not? Now, what do we actually need to predict severity? At the most basic level, we need data. Ideally, this would be molecular data such as proteins, metabolites, lipids, transcripts, or some clinical marker that can be measured in patients. We will need biological cohorts. We would need data, existing data from severe patients and non-severe COVID patients, as well as controls. We would need this data in humans. This is a severity prediction. So for other types of research questions, you can also go for animal models or cell line in vitro data. But in this particular case, it makes sense for us to gather this data in humans. And ideally, we would need a sample that can be collected in the clinical setting easily, such as blood, urine, saliva, or a throat swab. A tissue biopsy sample, for example, here would not be the most practical one, since you can't do that widely on a large number of patients. Before we go into a live demonstration of the platform, I would actually like to take a few minutes to walk through how you would normally go about solving this problem. So once you've identified what type of data you need and what is the research question you're asking, there are two options that you can start from. Option one is you can design your own study and collect your own data. And option two, you can find already published data sets or legacy data sets, and you can reanalyze them. If you go the first route, you would need to actually collect this data, pre-process it, and analyze it. You would then still need to go and do a literature search and find other data sets published by other research teams and compare your results with those. In both of these cases, you would synthesize your findings, you would generate new hypotheses, and there would be a research outcome, whether that's a new publication, whether those are further experiments, or if you're very lucky, you could be straight away going to trials. If we do go the first route, which is designing your own study and collecting your own data, it does involve very significant time and cost commitment. You would actually have to partner with a hospital or somewhere where COVID-19 patients are. You would have to get regulatory clearances, and you would need skill sets in your team that can analyze multiple different types of data sets if you're collecting different omics data. So for example, skill sets required to process a metabolomics data are very different from those required to process a transcriptomics data or a single cell data. If you go the second route, which is to start with data sets that are already out there, and for COVID, as well as for a lot of other disease areas, despite the best intentions of the research community, the data are available, but they are scattered across various data management systems, data repositories, publications, and so on. So let's look at an example of one publication. So this was a publication that recently came out in Cell in July 2020. This is one of the data sets that we are going to talk about later in the demo. So in this data set, proteomics and metabolomics data sets were collected for in blood serum of patients with COVID-19. The processed proteomics and metabolomics data are available as Excel sheets in the supplementary information of the paper. Raw data from the paper are available in a public data repository for proteomics data, integrated proteome resources, and code for analysis is available in GitHub. So if you actually wanted to use these data, you would have to gather this data from all of these different sources and analyze it just like how the authors analyzed it, or use your own data pre-processing pipelines. And this problem is not unique to this particular publication. For COV2, when I did a basic search, there are about 77 studies available in Gene Expression Omnibus, which is a data repository for transcriptomics data that contains bulk RNA-seq and single-cell data sets. There are 55 proteomics and metabolomics data sets in Massive. There may be more. This is just what showed up. And many, many more data sets across different repositories and different publications. In addition to these, you may have collected your own data sets, or other research groups may have collected their own data sets that are not made public, that may be in their cloud platform on their local systems and their local data management systems or electronic notebooks. In addition to data being scattered across various different sources, the metadata associated with those experiments are also often inconsistent and limit their utility. Legacy data sets, whether you have generated them on your own or whether others have generated, often require hours to days of work to clean up for even a single data set. So on your screen is an example here from Gene Expression Omnibus, where the expression matrix is actually split into 3 different files. In addition, even on the same platform, you can see at the bottom that the gene identifiers used are inconsistent. So we know from experience, as well as from reports of other researchers, that scientists spend hours and weeks, depending on how many data sets that they have to process, to make them analysis ready if there are not your own data sets. And only 3% of data sets in Gene Expression Omnibus, which contains approximately 60 to 70,000 data sets, are machine readable. So if you actually want to build machine learning models on top of the data, the work that you would require to clean up this data will just shoot up exponentially. In addition, metadata is often not standardized. So metadata standardization is actually not the norm, it's the exception. So it's not just making a data set analysis ready. Once you find it, often you will actually not be able to find the most relevant data set to your research question. So on the left are examples of 3 different studies from Gene Expression Omnibus, and you can see different conventions for indicating biological and technical replicates. So a human can potentially understand these, but to build machine learning models, these data will just not able to be, we will not be able to use these data because the identifiers and conventions for naming them are not consistent. So use of controlled vocabularies or ontologies is very, very rare, and it's extremely patchy when present. And if you want to find a very specific set of data sets, for example, type 2 diabetes studies where a particular gene is differentially expressed, it can require anywhere from days to weeks just to locate those data sets. And we're not counting the time that you would actually then need to clean up those data. So what Poly does is solve this problem of availability not being equal to usability. So on Poly, we are bringing data from multiple different sources together, and we're doing it in a very intelligent way. And I'll explain later on what that means. So we are bringing in data from different omics processing softwares, from different file storage systems, from databases, whether they are public or your proprietary databases, from LIMS systems, from electronic notebooks, as well as from publications. We're also bringing in already curated repositories that are present on Poly, and we're combining them to build enterprise level atlases. So these are your custom data lakes that can be used to answer a research question of your interest. And you will hear a lot more about this in other talks in the Poly UGM. Now, Poly not only brings all of this data together from different sources, but it also makes data machine actionable. What that means is on Poly, all of the data sets are stored in a very consistent file format, and metadata associated with samples as well as experiments are standardized through the use of ontologies. So all of this data is available to you in one place, and it's available to you in an analysis ready manner. So with this, we're going to actually jump to the live demonstration of the Poly platform, and I'm going to show you what a data lake on Poly looks like. When you log into Poly, this is the screen you're going to see. So there are three different sections: workspaces, applications, and discover. Discover is where all of the data on Poly lives. So I'm not going to talk about workspaces and applications today. We're going to jump straight into discover. So as Discover loads, if you have access to several different data lakes, you will see a number of cards here. All of these, every single card is a data lake. So today we're going to talk about COVID-19 multi-omics data lake. So we're going to jump straight into that. Now, a data lake on Poly can have access to a single type of data or multiple types of omics data. There are two major parts of a data lake. On the left, you are seeing a filter screen with several fields that you can use to filter a data set and find a data set of interest. On the right, you will see a list of all the data sets that are present in your data lake. So in this particular data lake, there are currently 80 data sets, and as more data sets are added, this number will get updated. So since our research question of interest today is to predict COVID severity, we're going to take a look at how many COVID data sets are present here, how many of those are human samples, and how many of those are in tissues that can be easily measured or collected in a clinical setting. So if we filter by COVID-19 disease, we will see that there are 30 data sets. We can further filter by Homo sapiens as an organism, and we now have 26 data sets. So we can see there are lipidomics, metabolomics, proteomics, single cell, and bulk RNA-seq data here. So we are going to look at proteomics and metabolomics data today. So there are three proteomics data. I'm going to choose one of them. Now, before we dive into analysis of these data, let me also walk you through what each of these columns are. So each of these columns here have come from that intelligent processing of different data from different sources that I talked about. So Poly takes these data from different repositories, from publications, standardizes the metadata, standardizes the format of these data, and then puts these data in an analysis ready format here behind the data lake. So that's what we are seeing. All of these fields have been curated with a semi-automated process. So we have machine learning models that help us tag these different fields with ontologies. And then there are experts that oversee that process and correct any errors in the automated models. So in the for each data set, there is a unique ID that you can see here. If the data set is coming from a publication, it will be the name of the publication and the authors. If it's coming from a public data repository, you will see an ID of that repository. Like here, you can see a gene expression omnibus ID. You'll see a brief description of the data here, which will tell you what that data set is about. You'll see different fields like organism, type of data, disease, tissue, and you'll see a link out to the source. So if you click on these links, you will be taken to the relevant publication or the relevant repository where these data are coming from. Once you select a data set, as we have done here, we've selected this proteomics data, which is about large-scale multi-omic analysis of COVID-19 severity. You will see a pop-up open at the bottom here. From this drop-down, you can select where you want to open these data and how you want to analyze them. These options will change depending on the type of data you select. But there will be two main types of options. So there will be point and click solutions and there will be notebooks. So if you are a computational biologist, you may prefer to open these data in a poly notebook where you can write code and do your analyses. Or if you are a biologist, you may prefer to open it in an application, which is more point and click. So you will see both of those options here. So there are some pre-built applications that come pre-installed with Poly, but additional applications, your favorite tools can also be added here. So you don't have to learn a new tool and can continue to use what you are familiar with and experienced with. So here we will first open these data with a point and click solution. So it's an application called Discover. I will select the application and I will click on Launch App. When I do that, Poly will ask me to select a workspace where I want to save my analyses. All of the analyses on Poly are saved and versioned, whether you are in a point and click solution or you're using a notebook. Therefore, I'm selecting a workspace. I can give a custom name to my analysis. For example, COVID-19 severity proteomics in plasma. So once I'm happy with the name, I can click launch and the application will open in a new screen. So it'll take a few seconds to launch. While the application is launching, I want to go back to the presentation and take a few minutes to talk about what these data are and what we expect to see today. We're going to use Poly data lakes to answer 4 different questions. We are going to see what proteomic markers of COVID-19 infection are. We're going to look at a metabolomics data set to answer what metabolomic markers of COVID-19 infection are. We're also going to look at how consistent these markers are across studies. So we're not just looking at one data set, but also several different data sets in a data lake. And then we're going to run a machine learning model and ask the question, can these markers be used to predict the severity of COVID-19 and what is the accuracy of that prediction? There are 2 main data sets that we're going to talk about. One is a proteomic and metabolomic characterization of COVID-19 patients in blood serum. So in this particular data, there were 28 healthy subjects, 25 non-COVID-19 patients who had clinical symptoms severe to COVID-19 but tested negative. And then there were 37 non-severe patients and 28 severe COVID-19 patients. And severity in this study was defined based on clinical criteria of respiratory distress, blood oxygen saturation levels, and oxygen partial pressure in the blood. So a very interesting study, same cohort of patients, 2 different types of omics data were collected. Another study that we are going to talk about today also came out recently, and it's a large-scale multi-omic analysis of COVID-19 severity. So here, 4 different types of data were collected on the same set of patients. So there were 26 COVID-negative patients and 26 COVID-negative patients and 103 COVID-positive patients. The authors collected bulk RNA-seq data, proteomics, metabolomics, and lipidomics data from the same set of patients. Another very interesting thing about this data was that instead of using clinical characteristics like oxygen saturation levels, they used a metric of hospital stays. So the authors said that if the number of hospital-free days was less than 26, the patients were severe. And if the number of hospital-free days was greater than 26 as measured on day 45, the patients were non-severe. So 2 different studies, 2 different measures of disease severity. This is a data set that we're going to look at in poly in a second. But before we do that, I want to highlight that some of the key biological processes that this particular publication identified as being dysregulated were lipid transport, blood coagulation, acute phase response, endotheliopathy, and neutrophil degranulation. And we are going to take the proteomics data from these studies, open it in an application on poly, and see if we can recover some of these biological findings that this paper has outlined. Once the application opens, you will see a screen with an overview of what the application does. You can take a look here, and you can also go to the help section of the application to get more information. Now, I'm not going to go into detail of what all of these different tabs are. I'm only going to highlight certain key aspects of the biology that are important for us to understand the molecular markers of COVID-19 severity. So you can scroll through different options available in the app on the left here. First of all, we can take a look at all of the different types of metadata that are available for each of the samples. So each row in this table is a sample, and you can see all of the clinical measurements that the authors had taken in the study here. You can also see options that Polly has added of disease and disease type, control vocabularies here. And you can take a look and scroll through them. So here we have data for 129 patients for non-COVID-19 and COVID-19 disease of different severity. You can do data set level statistical analysis, such as a principal component analysis plot. And you can see here that the severe and non-severe COVID-19, some of the samples are segregated quite well, but others have looked very similar, at least at a data set level, to the non-COVID-19 patients. So we definitely need to do more detailed statistical analysis to pick up the biological differences between these cohorts. We can take a look at the box plot of different samples to see that the data set are already normalized. We can also go and visualize different genes and different genes in different pathways here as bar plots and as box and whisker plots. So here I've plotted 2 proteins, SAA1 and SAA2, and you can see that these are definitely high in severe COVID-19 patients, and they have been implicated as one of the major factors correlated to severity, and you can see the same thing in the box and whisker plot, as well as the heat map of average protein expression here. Now, let's go to differential expression. And here I am comparing non-COVID-19 biological cohorts in these data, which are non-severe non-COVID-19 and severe COVID-19, with the severe COVID-19 patients. And these are the results of these analyses will give me which proteins are differentially expressed in severe COVID-19 and can be potential markers. So I'm selecting some parameters, my p-value cutoff, as well as my log2 fold change cutoff for my differential analyses. I can run these analyses, and I can see a list of proteins in this volcano plot. and I can scroll through them and go through them one by one and see what's upregulated and what's downregulated. And I can also see these results in a table. And if you look at the top negative fold change protein, that's CRTAC1, which is one of the proteins that the authors in the study also highlighted. You also see ApoA2 showing up here, which is a lipoprotein, kind of indicating and hinting towards dysregulation of lipids in COVID-19 infection. Now, what is more interesting here is not to look at a table and different proteins, but to go and look at what these changes actually mean. So I'm going to go to pathway analyses, and within the pathway analyses, I'm going to look at gene ontology. So that is one of the fastest ways for me to pick up which biological processes are changing most significantly between severe COVID-19 and non-COVID-19 cohorts. So here I've chosen my parameters for this test, and I am doing the gene ontology of biological processes. And when I run this analysis, straight away, literally within a few minutes of me opening this data set, I can see that platelet degranulation is dysregulated. I can see acute inflammatory response, which is to be expected for any viral infection. I can see blood coagulation as significantly dysregulated. Again, several studies as well as clinical observations have identified increased clot formation in patients with COVID-19, and we are seeing that in proteomics data here. Similarly, there's negative regulation of peptidases showing up here, something that the authors didn't quite talk about, which could be a potential new hypothesis that we can look at. We also see lipoprotein particle clearance showing up, again hinting towards lipid dysregulation. Again, hemostasis showing up, blood coagulation pathway showing up. Now, one of the other important biological process that this study pointed to was neutrophil degranulation, and we see that showing up here very clearly. And again, several pathways related to neutrophils are showing up. So as you can see, there was this study that was published. The data is present on Poly in a curated fashion and analysis ready fashion. And it literally took us a few minutes to go from there to biological interpretation of these data. And now we can take these data and form our own new hypotheses and analyze these in a different way if we want. Now, before we go and do that and we compare different proteomics data sets in our data lake, I also want to go and look at metabolomics data briefly to show what are the, to see what are the metabolic markers of COVID-19 infection. So I'm going to go back to my COVID-19 data lake. And I will find a metabolomics data, and this time I'm picking the second data set where the authors had measured proteomics and metabolomics on a cohort of healthy non-COVID-19 and severe and non-severe COVID-19 patients. So the process of opening a data set is the same. I will select a data set, and from the drop down, I will select which application I want to open these data in. Now this, as you can see, this is dynamic. So when we had proteomics data, we opened it in an application that was compatible with proteomics data. And here I'm seeing an application for metabolomics data. So I'll select the application, I will launch it. I will select a workspace where I want to save my analyses, and I will open my analyses. So again, this will take a few seconds to open. I have the data open already in a different tab. So we'll go and start analyzing these data there. Now, one of the key biological processes that the authors had identified in these data was amino acid dysregulation. And you'll see very quickly how we can arrive at the same conclusion in a few minutes. So the first thing we can do, just like we did with proteomics data, is look at all of the metadata available here, and we can see patient ID, disease type, an annotation Poly has added, gender and age. We can look at the distribution of densities for the metabolomics data, and these plots are telling us that the data are normalized, so they are ready to analyze. Just like we did for proteomics, we can also do experiment level statistical analysis. So we can look at the principal component analysis for the whole data, where we can see that the healthy and the non-COVID-19 patients are quite well segregated from the COVID-19 patients. But the distinction between severe and non-severe patients is not quite clear, which is where the need for machine learning models comes in, as we will see later. Now, we can go and do a differential expression analysis between healthy and severe COVID-19 patients. We can again look at the results in a volcano plot. We can look at the table and look at the metabolites that are that have a high negative fold change and that have a high positive fold change. But what is more interesting again is to look at enrichment analysis, which will tell us functionally which pathways are changing. And if we look here, it's just very, very obvious that amino acid metabolism is very fundamentally changed. So you will see several amino acid metabolism pathways show up here. We see valine, leucine, and isoleucine, see taurine and hypotaurine, glycine, serine, and threonine. Again, this was one of the major takeaways from the study as well. You also see indications of lipid metabolism show up here, which we also saw with proteomics data. So sphingolipid metabolism is changing. So are some of the others that again point us in interesting directions. These are threads that we can explore and test further. So as I said, I'm not going to go into a lot of detail of all of these tabs. You can reach out to us if you want to learn about these in more detail, or you can, I will point you to resources where you can read more about them. Now, just like we did with proteins, we can also take a look at what metabolites are changing. So some of the metabolites that showed up in our analyses in our differential expression were related to sphingolipid metabolism, and we can see in severe COVID-19 patients, sphingosine and sphingosine phosphate are downregulated. Ocresol sulfate was one of the metabolites that had the highest negative fold change of -0.6, log2 fold change of -0.6. So that's very, very profound. And kynurenine was another metabolite that showed up in the study and has been associated with COVID-19 severity. So you can do these comparisons one by one and kind of try to identify what is changing in severe patients versus non-severe patients. But you can also compare what are the common markers across different data sets. So one data set can only tell you so much. The power of a data lake really comes into full visibility when you start comparing different data sets together. And one of the other things that is often implicit in how we work with these analyses is it's very rarely one person answering these questions. It's often a team of people of different skill sets that are working together. So that is exactly how our team at Elucidator works as well. There are several people who work together to answer these research questions. And Poly is the platform where we collaborate. So all of the analyses that I have done, these analyses get versioned and saved in Poly, and my colleagues can actually go and look at them and see what analyses I have done, and they can look at the reports that I've generated. In the same way, I have data scientists in my lab who actually run machine learning models on these data, and I can look at their results in real time when they're doing the analyses. So with this, I want to go to the third question that we highlighted, and I want to look at some of these markers that we just showed, we just looked at in a data set. How consistent are these across data sets? And one of my colleagues actually did these analyses and shared those with me on Poly, and that is what I'm going to show you now. So as I had showed you earlier, you can either do the analyses in a point-and-click fashion in an application, or you can actually open these in a notebook and write your own code. So my colleague is a data scientist and he prefers to do his analyses in a notebook. So to look at analyses of a colleague, I can actually go to my workspaces and I can filter for the notebooks that my colleague has shared with me. And I can see that these are all of the notebooks that he had generated. And one of the main analyses that he had done that I want to share with you is whether these markers that we have seen of COVID-19 severity are those common across different diseases. What Poly does is, in addition to all of these point and click solutions that I showed you today, there is a very powerful computational infrastructure running behind as well. So for very heavy computational analyses, you can choose machine configurations with very high RAM and CPU, and you can write your code and essentially scale your computation. For example, this notebook was last modified. Today, I can see all of the activity. I can see all of the versions of this notebook as well. And when I open it, you will see that I will be able to access my colleague's analysis. In this particular analysis, my colleague got the analysis data sets directly from the data lake. And they did a differential analysis of protein abundance of severe COVID-19 with non-COVID-19 patients in both the data sets that I talked about today. So two proteomics data sets coming from very different patient populations with different severity criteria. We want to see what is, whether these markers of severity are consistent across these two data sets. Again, this analysis can easily be expanded to more data sets from a data lake as well. So these analyses were done in a Poly notebook environment. For people who code, this may be very familiar. And you can write code in any language that you prefer here, for example, R, Python, or a mixture of the two. So I'm not going to, again, go into details of how to use a notebook, but I'll just point to key biological findings of these comparisons. So we went ahead and downloaded the data sets from our data lake. We did a differential analysis of severe COVID-19 with non-COVID-19 patients in each of the two data sets. Now, this is pretty simple again, because the samples for each data are pre-annotated. So when you look at the data, you know exactly how many samples of each type there are. You can make your own custom cohort and design your own analyses. So once these were done, you can see in the first data set, which was a proteomic and metabolomic study, there were 20 upregulated proteins and 14 downregulated proteins. We did the same thing with the second multi-omic data, and we can see that there are 51 downregulated proteins and 104 upregulated proteins. And now that we have these markers from both of these data sets, we can ask the question, how many of these are common? And when we do that, we can see that there are 9 proteins that are upregulated in blood plasma in one study and blood serum in the second study. And when we look at these upregulated proteins, this is something very, very interesting. So what you see here, as expected, a lot of complement proteins, complement pathway proteins show up, as you would expect. A lot of coagulation factors show up, are upregulated. Very interesting gene that shows up here is ORM2, and it's not quite clear what the function of this gene is, but it is upregulated in both of the proteomics data coming from very different patient cohorts. So this is possibly a place where we can delve into the data further and try and identify what ORM2 is linked to. Same way, we can look at the downregulated genes as well that are common in both studies, and there are 6 downregulated proteins that are common. There is the A2M protein. We know that that inhibits inflammatory cytokines in the body, and it is downregulated. So it is an indication of the acute inflammatory response that is happening in severe patients that is not happening in non-COVID-19 patients. You see ApoH show up again, which is an important component of lipid metabolism, but it is also again linked to coagulation pathways in the body. Another interesting protein that showed up is very highly downregulated is CRTC1. And we saw this in the proteomics analysis as well, that this is a protein that had a fold change, log2 fold change of -6, and it is showing up in the second proteomics data as well. And it is not quite clear how this is connected to COVID-19, so another potential area of exploration. So now that we are starting to put multiple data sets together, we start getting this list of prioritized genes, these smaller lists that are showing up across multiple diseases, and they increase our chances of these markers being real for COVID-19, the more data sets we expand these analyses to. So this is where I'm going to leave the thread of comparing different data sets together and go to a machine learning model and how we can build a machine learning model and use that to identify markers of severity. So for this, I'm going to open another notebook. And again, this analysis was done by a colleague of mine and shared with me on the Poly platform, and that's what I'm showing on my screen right now. So the major questions that we asked for this analysis was, can we use the omics data in Poly data lakes to build models that will classify COVID patients into severe and non-severe? And the intention behind it was what we had articulated at the beginning of this session, is can we predict which patients are going to become severe so that we can optimize their care and hopefully improve their outcomes. So what we're doing here is we are fetching the processed proteomics and metabolomics data from one of the studies, and then we are building a model for severity prediction. Again, doing this is literally a few minutes job because the data are already cleaned, the metadata already have tags. So I'm not going to go into exact details of how the model was built. Suffice to say, we got the data from the data lake, posted proteomics and the metabolomics on the same set of patients. These data sets, both proteomics and metabolomics, were put together. And we ran, again, we did differential expression on these data. And here, one of the key differences from all of the analyses that I showed you earlier was here we are comparing non-severe COVID-19 patients to severe COVID-19 patients, because these are the cohorts that we're trying to differentiate from. So we're not looking for markers of COVID-19 infection. We are looking for exactly what differentiates a non-severe case from a severe case. So you can see differential expression of severe versus non-severe. There are not a lot of proteins that are different there. There are actually 16 downregulated proteins and 25 upregulated proteins. And similarly, we can do an analysis for metabolomics data. And we can see, again, not a lot of difference, but there are some key metabolites that show up. And you can see there are 5 downregulated metabolites, many lipids among those, as you can see here. A lot of glycerophospholipids are showing up here. We see kynurenate and kynurenerin show up, which we had identified earlier. And we also see, again, sphingolipids show up as markers, actually as differentiators between severe and non-severe COVID-19. Now we can take these and actually build a machine learning model on top, and that is exactly what we have done. So I'm going to scroll through these and kind of just walk you through this random forest model that we had built, and we are ranking exactly the factors and identifying the most important predictors that differentiate these 2 patient cohorts. And these are the top patients. Again, this is a model that combines 2 different omics data types together. We can add more data types as well, but as you will see, this model is already pretty accurate. So we can check how well the receiver operating curve looks for this model, and the area under the curve is close to 0.96, which is pretty good for a model. Now, to test whether this model works well or not, we actually ran this model on a test cohort, and you can see the results here. And we can see that one of the severe patients is incorrectly predicted, which is exactly what the authors had also found in the model that they had developed in the paper. So what we see here is that the data set was present on Poly in an analysis ready manner in a machine readable format. So we can actually take that data and build an ML model in literally a few minutes. And we can reproduce the results that a published study got. And this is just the beginning. So we can also now start including other omics data as well as data from other studies in our models and try to refine them and improve our predictions. And here you can see a side-by-side comparison of the result we got from Poly and the result that was in the publication. So to conclude, Poly provides access to machine actionable multi-omics data. We saw that today with the data lakes, where different kinds of omics data can be present in one place. And these data sets are tagged with control vocabularies, and they're already made analysis ready, so you can build ML models on top. These data are also linked to a library of customizable and dynamic analytical tools. So each type of data is linked to a tool that data is compatible with, and these tools are customizable. And again, I invite you to attend a talk by my colleague Sudhi Poddar, where she will talk about how you can customize these tools, how you can add your own third-party tool or build a custom workflow on Poly. Now, the tools that are on the platform are designed keeping in mind an interdisciplinary research team. So we recognize that often the teams working on research problems don't just consist of biologists or computational biologists, but these two different skill sets working together. Therefore, we have both point and click solutions as well as tools for more heavy computational analysis, as well as a very robust computational infrastructure behind the platform, where you can access our platform through APIs, through command line interfaces, and write your own code and add your own tools as well. Now, we are also continuously adding new data to Poly. We saw COVID-19 multi-omics data lake today, but we have several others on the platform, and we are adding new data repositories every single week. I invite you to attend a talk by my colleague Shwetab, where he will talk about the roadmap and what we have in store for you in the future on Poly. I also invite you to check out a COVID-19 Omics resource that we will be releasing at this user group meeting. The link to the resource is given here. So the data sets that I talked about today and several other data sets will be available in this omics resource for you to view. I also want to give a shout out to some of our users and scientists who will be presenting their work today. I particularly want to highlight Dr. Thakli's talk on integrating Poly into their lab's research process. Also talks on how Poly is being used for multi-omics integration of early stage drug discovery, as well as how Poly enables analysis of tracer metabolomics. So these are some examples of how our users today are using the platform. And with that, I would like to thank all of you for giving me time, taking the time to sit through this demonstration. And if you have any questions, feel free to contact us or visit us on our website. Thank you.